Good morning. We are going to go ahead and get started. Can I have your attention, please? Time to get class going. This is very empty. I'm going to have to send an email and trying to get more of you guys to show up. But thank you for being here when the weather is not great. This is one of the more intensely dense classes of this course. We're going to cover a lot of material. So it's really good that you're here in person because I think that'll help you understand it. Okay, so before I get into the class material, just another plug for this. Today is Pi Day 3.14, March 14th. If anybody is familiar with the mathematics fun that Pi Day brings. So it's a good day to start this cookies and conversation thing with STEM faculty. I will be in the Student Science Learning Center, Math and Science Learning Center, 2 floors up at 4:30 p.m. With cookies, ready to talk to you. Hey guys, please pay attention. Thank you. So I will be there in the afternoon. I hope to see some of you. Tell your friends this is not a cell bio specific thing. This is something we're trying to do across STEM, maths, chemistry, biology, physics. So please tell your friends to come see us. If not today, then Stephanie Colonia a week from now, and then Alexandra Naba on the 4th, and so on. Okay. So with that, we will continue where we left off last time with the cell biology. And where I left off was covering positive and negative feedback. All right. So just to recap, because it is where we left off, it was the very end of class. If you have a signaling pathway that has positive feedback, then what's happening is that X is activating in some way Y, which again, as I mentioned last time, you don't know what's happening. This doesn't tell you if you're making more of Y or making Y more active, but one way or the other, your Y is doing more stuff, whatever that might be, downstream of Y. And in a positive feedback loop, as you make more Y and it does whatever it needs to do, one of the other things it does do is to go back and tell X to make more, or rather activate more of X, or make more X. And that in turn makes more Y. So if you were to chart the progression of the amount or activity of Y over time, it would do something like this, where it just went up really fast. In a negative feedback loop, it's the opposite, and the end result is a little bit more complicated than just a curve that goes in one direction. If you have Y being made, and it's doing its job, it's also sending feedback saying X, let's make less X, or make X less active. And since X's job is to make Y active, that's going to make Y less active. As Y becomes less active, it can't do this feedback as well, and X will eventually start to go back up. And as it goes back up, it will activate more Y. And so you end up with this sort of back and forth thing going on that can really, it's very dependent on what else is coming into the signaling pathway. So I guess you could think about this as the accelerator is floored down and the car is just accelerating with no end in sight. And you can think of this one as sort of somebody who's like a really terrible, so basically a typical Chicago driver, all right? You know, like accelerate, brake, keep going back and forth. There you go. So that's what that is. Race car, Chicago driver. Okay. So I'm going to skip these next 2 slides, this one and this one. They were simply examples of how complicated signaling is. And for time and to catch up a little bit, I'm just going to skip them so you don't have to worry about them. If you're interested, you can just read about this on your own. It's just sort of a nice example of how complicated signaling pathways are, but we're not going to cover that right now. Okay, so moving forward then, we talked about last time about ligands, the big category of things that bind to receptors. We talked about receptors and their downstream signaling components. These are some of the terms we discussed, signal transduction proteins. To transduce something means to relay it. If you're running a relay race, right, you might have a baton and you're going to send it to the next person and the next person and the next person. That relay race is a lot like what's happening in signaling, because in one of these pathways here, you've got relay runner 1, 2, 3, 4, right? That's kind of the same idea. You're taking the signal and you're passing it along to the next component to take it somewhere else. All right, so signal transduction proteins are proteins that relay the signal, that transduce the signal. Intracellular signaling proteins, that just means intra means within. So inside of a cell, you have signaling proteins. Not a lot of information there. And these 2 terms are often used interchangeably because any protein that's inside of a cell signaling is usually, by definition, doing this. It's kind of the same thing. Second messengers tend to refer to proteins or molecules that are further down the pathway that lead to effector molecules that are named that way because they cause the end effect. And as I mentioned last time, what is the end effect is a very semantic, difficult thing to describe. And that's why we're not going to spend a lot of time on these terms, but I want you to just be generally aware of them because they tend to come up a lot as parts of questions in your entrance exams and so on. If you see these, I don't want you to go, what on earth is that? Never learn what a second messenger is. Just understand a second messenger is part of the signaling pathway inside of a cell, and it's carrying the signal somewhere from up here to somewhere down at the very end of what it does. Okay? Effector molecules are usually proteins. Examples, kinases, phosphatases, G proteins. We're going to cover a lot of these, but these are some examples of effector molecules. So a really important point we're going to talk about now, some intracellular signaling proteins act as molecular switches. And the term switch is really appropriate. You've heard me last time mention the idea of turning a light on and off, and that idea applies here too. So you can kind of envision in your mind a switch, a molecular switch, where it's turning things on and off just like a light switch would. And so let's talk about what this molecular switch idea means. Many of these signaling proteins act as molecular switches, alternating between an active, or quote-unquote on state, and an inactive, quote-unquote off state. Active and inactive are better terms because on and off is a little too simplistic, as you'll start to see as we go through the next few slides and lectures. But, and in fact, I added this because this is the important point. Or one of the important points. If you're in a room and you turn the lights off, I think I mentioned this last time, obviously lights on is more useful than lights off. That's where the analogy breaks down here. When you're talking about signaling, both the active and inactive states are equally important for the signaling process. Right? So whereas you couldn't do anything in the dark, if you turn a signal off here, the absence of that signal still means something to the cell. And it can still do something with that information. So that's sort of the difference there. These are the two very common molecular switches that we'll cover over and over and over in the next few lectures. I sometimes tell students, if you're ever in doubt in life, just assume something is being phosphorylated or dephosphorylated. It's so common. So you've heard hopefully about the terms kinase and phosphatase from this class and other classes. A kinase is an enzyme. Remember, it ends in ASE, so it's an enzyme. A kinase is an enzyme that adds a phosphate group onto something else. That's all it is. A kinase adds a phosphate group. A phosphatase is an enzyme that takes a phosphate group off. So they do basically the opposite things. Kinase adds a phosphate, phosphatase removes it. Sorry, I'm fighting off a little bit of a thing, so if I cough, that's why. Okay, so let's say you have a protein here shown in blue, and it's quote-unquote off, it's inactive. A signal comes in through the signaling pathway, and it tells a kinase, which is, remember, one of these effector molecules, the end effectors that does something, and that signal coming through a relay chain of A, B, C, D, whatever, it now is telling the kinase, add that phosphate group onto this protein. When that phosphate group is added, it turns the protein, quote-unquote, on. It makes it active. All right? And the last time I told you about shape changes, conformation changes, assume that's what's happening here. And almost, in fact, in any case that I know of, the reason this gets turned on is because that phosphate group takes that protein and it causes it to change the way it looks a little bit. And that change of the way it looks changes what it does, whether because maybe it makes a new site on the protein available, maybe it hides a different part of the protein, whatever it does, it turns the protein on. So I was telling somebody in office hours today that if you looked at a light switch and you just have it in a down position and you move to the up position, that shape has technically changed, right? It has a different look to you. Same idea here. The protein is changing and then it's doing something. All right. So that will turn it on, and the phosphatase then does the opposite. It can take the phosphate group off and turn the protein back off. Now, there are exceptions to this. Usually, not always, but usually a phosphate group addition turns a protein on, makes it active. But as you'll see, actually, even in this class down the road, there are some exceptions to that. Sometimes the phosphate group can turn it off, and then the removal turns it back on. But in most cases, this is how it works. The other example that we'll cover a ton is signaling by GTP binding proteins. Now, you are probably much more familiar with ATP, adenosine triphosphate, energy source. You've heard about it a ton in all your bio classes. You've hopefully also heard about GTP, which is guanosine triphosphate. So using these nucleotides, adenine and guanine, you're making these molecules. Guanosine triphosphate, 3 phosphate groups, just like ATP has 3 phosphate groups. Guanosine triphosphate is at the root of this GTP binding-based signaling pathway. So in this example, what's happening here is you have a protein in green that's off when it's bound to guanosine diphosphate, 2 phosphate groups, not 3. And then a signal comes in, and while it's not shown here what's doing it directly, what that signal does is it takes this GDP out. Sorry, it takes this GDP out and puts a GTP back in. Now notice how this is different from what I just described to you here in a very fundamental way. I told you here that you're taking an existing protein and simply modifying it with a phosphate group, right? That's not what's happening here. It's not that you're adding a phosphate group to the GDP to turn it into GTP. You could in theory do that, but that's not what's happening. Instead, you're actually removing the whole molecule that's got 2 phosphates and putting in a different molecule that's already got 3 phosphates. So the end result is the same. It's done differently. It's sort of like here, I'm wearing a hat, and because I'm feeling, I don't know, fanciful or something, I put a flower in my hat or a feather in my hat, and now it's all stylish or whatever. That's what's happening with the phosphate group here. I'm adding something to the existing hat. Here, though, I'm taking the hat off and putting a different one on, and that different hat already has the feather on it. All right? So it's sort of, at the end of the day, if you step back and say, what's the same thing here? Well, you have an extra phosphate here than you did here. You have an extra phosphate here than you did here. But how you got to that is very different. Yes? So the question is, does that have something to do with the activation energy? Sort of, yes. That's getting a little bit outside the scope of this class, but it does require, on average, more energy to take another molecule and plug it in and take something out than it does to just put a phosphate group on. So you could make an argument that some of these things have evolved to work in certain ways based on certain energy levels. But that gets into both speculation and a little bit outside my area of expertise and way outside the scope of this class. But it's a very good question. Yeah. I saw another hand somewhere. No? Okay. And I have the candy again, so ask questions and come get candy afterwards. Okay. Right. So the second part of this here is more similar to what happened here. Okay? I just told you this is different than this. But removal of the phosphate group with this signaling pathway is pretty similar. You had a phosphatase here that took off the phosphate group. And here you have what's called hydrolysis, which you've hopefully learned about in chemistry. And that hydrolysis of GTP turns it back into GDP and releases the phosphate group. Okay? So here, a phosphatase enzyme took the group off and tossed it away. Here, somewhat similar, but not the same thing, but somewhat similar, hydrolysis took a phosphate group off and threw it away and turned it back into here. So to recap this very quickly then, the addition of a phosphate group, if you will, is not because you're adding it, it's because you're taking this out and putting a new molecule in. But the removal of that phosphate group to go back to this is just taking the phosphate group off. Yes?
Is it done by some type of enzyme, like GTPase?
Yes. Is this done by some type of enzyme? Absolutely, and that's what we're going to cover today. So there are enzymes, and you're exactly right, it's called a GTPase. So you're fantastic. You're thinking ahead. A enzyme ends in ASE, and whether you read ahead or just guessed, it's exactly right. It's called a GTPase. When you take that GTP and you pull a phosphate group off, and now you have a GDP. Sorry. So we'll get into that in a second. Okay. So I already kind of said this, but the on-off switch is more of a fork in the road. I want to re-emphasize that because the off position, the different level, leads to different states. So that even here when this is off, it has an effect in terms of what happens in the cell. It's not like it just goes away or something, right? The protein being off can also have an effect. All right, so now we're going to talk about very quickly an overview of 3 types of receptors. In the next couple of classes, we'll cover those receptors in detail. So there are 3 main classes that we will go over, and this is all in chapter, I guess, 16, I think is where we are in your book. I hope, yeah. Ion channel, yeah, it's chapter 16. Ion channel coupled receptors, G-protein coupled receptors, and enzyme coupled receptors. This one is super straightforward. We're going to spend like 2 slides on this basically. We're going to spend the rest of the class and maybe some of next class on this one, and then the following class on, and the next class on these guys here. So at the moment I'm not going to go into detail here because we are going to cover them, but this is an overview slide that you can come back to and say, okay, these are the 3 receptors I need to know. Very quickly, the ligands for these 4 types, or I mean these 3 types of receptors and a 4th one here, work in slightly different ways. And so I guess I will give you a bit of an overview of that right now. So the first type of receptor, a ligand-gated ion channel, which I think Dr. Lin's already told you a little bit about. In this case, an agonist. Remember, an agonist is a type of ligand. An agonist is a ligand that has an effect. So it makes sense to talk about agonists here. Agonists, in this case, will encourage a ion channel to open up and to let ions such as sodium, Na, flow through, and that can have downstream effects. Super simple concept. In this case, with a G protein coupled receptor, sometimes also called a 7-transmembrane receptor. It's called that because if you were to count these wiggly lines, they go through here 7 times. It's a 7-transmembrane receptor. In this class, we'll call them GPCRs, G protein coupled receptors, but just be aware that this is a different name. Now, when an agonist binds to here, you have activation of what's called a G protein, and then signaling downstream of that actually makes use of a lot of what we just covered here, and that's what we'll talk about today. And then this one, enzyme link receptors, we'll cover next time. And then nuclear receptors I briefly mentioned to you. Sometimes agonists can actually cross the membrane, go inside of a cell, bind to a receptor, go into the nucleus, and do what they need to do. All right? So one of the takeaway messages of these series of classes is that when I show you a diagram like this, you don't need to memorize it. What you need to do is learn it. This goes back to how much time I spend on pathways. The arrows, upside down T's, inhibition of inhibition, activation, and so on. It all falls downstream of these ligands and receptors and so on. So you need to be able to learn to read this, and this is a good practice slide for you to do so. Ask yourself questions like, okay, what does it mean if I have a written here a protein kinase A, PRKA, with an arrow pointing to this big arrow? What that means is that this protein kinase A, this is just an example, is facilitating or moving or encouraging the reaction to move in this direction. And it's doing so by taking adenosine triphosphate and taking a phosphate group off of it to make this ADP, diphosphate, and adding it to a different protein, which is now activated, and then it does its job, and it happens to be a kinase as well. So these pathways work sort of like a waterfall cascade, going back to the first picture I showed you about the kayaker on the rapids. So think about them that way. Kinases can phosphorylate kinases, could phosphorylate a phosphorylase that then removes a phosphate, and so on and so forth. All right? So just, this is a good practice slide to play with. And to go back to the one I then ended on last time, when you can read these types of diagrams, you can learn all sorts of things, such as what different things inside of your body are doing. Okay, so with that, we will now go to the slides for today. Any questions while I pull up the next slide set? Yeah. Yes is the short answer. So the question was, when you take GDP and turn it into GTP, is there a conformation change in the GTP? So technically, yes, because GDP is this nucleotide-based molecule, and you've added a phosphate group, so you've literally changed what it is. What happens next, though, is the critical part, because, of course, GTP is not a protein. So what the critical part is that the GTP, as opposed to the DP, now binds differently to whatever it's targeting, and that causes conformational changes in proteins that then have an effect. Okay, so obviously I was running late on slides, so this would have been the first slide you would have seen today previously on the hit show Cell Biology. And the reason I do this, other than my dad jokes, is because I try to put in a few slides from the previous lecture that kind of highlight the things we covered. So since for today I'm just going to very quickly go through them. I mean, obviously I just talked about this one. This is a reminder that without signals, cells die. This is a reminder and an important one for today that the signal molecule, the same signal molecule shown by this red dot here, can have different effects in different cells based on what happens downstream of the receptor. Right? So this is one of the reasons you have these complicated signaling pathways, because what it allows you to do is take a single signal and have different effects in different cells because they respond differently to that signal. This is something we covered last time about endocrine, paracrine, synaptic, contact-dependent signaling, so something that you should be familiar with. And then, of course, general types of receptors. Fast and slow signaling, something you will hear about more today. The idea that signals can have immediate effects on cells by altering existing proteins inside the cell, but they can also have more profound changes by changing the RNA and protein that's made in the cell. And that's important for basically making a major change. So maybe it's the difference between, me changing my shirt quickly versus getting a makeover or something like that, like they do on those reality shows, right? This one is fast and this one is slow. Okay. I'll pass on all the jokes I can make about my age and needing makeovers. Okay. So the flow, is it really passing on a joke if you say, anyway, meta joke, hashtag. Flow of information during chemical signaling. This is a summary slide I talked about last time. Signal is a ligand, binds to a receptor, causes signaling through these molecules, causes an effect by effector proteins, and these are some of the changes that can happen. All right. So now what we're going to focus on are, again, two types of receptors today, or however much we can get through. Ion channel coupled receptors and G protein coupled receptors. These guys and these guys. So as I promised, just two slides on ion channel coupled receptors. They're very straightforward. Here's an example where the receptor is doing nothing. It's closed. This is a gate, essentially. These ions are unable to get through. In this example here, this is a binding site for a signal, and the signal binds here, and that binding causes, you guessed it, a shape change. And a shape change, in this case, opens up this gate. Now, these ions, because there's more of them out here in this example than there are on this side, inside the cell, they can flow right through. So that's an ion channel coupled receptor. It's very simple and direct. You open the channel, things go through it. It's like opening a gate or the door to your house. And an example of that is shown here, where you've got sodium going this way, potassium is going this way, and the cell can use this for a number of important functions that I think we'll cover slowly over time and that you've heard about. Okay, active ion channels, this was passive, right? You open, things flow along the gradient that they already have. An active pump or an active ion channel works against the gradient. Sometimes you want more of something inside of you even though the gradient is going the opposite way. So for example, you might want more potassium inside of the cell, but the cell already has more potassium than the outside of the cell does. So if you open up a channel, it would just move out, and that would be the opposite of what you wanted. So instead what you do is you don't open up a passive channel. You use energy to force even more of the potassium to come into the cell through a pump. All right, so the sodium-potassium pump is one of the most common examples of this, where it moves these ions as a result of protein conformation changes, shape changes again, while transiently phosphorylated. So this is integrating some of the stuff you heard about. Phosphorylation is a switch. It changes what a protein does, changes its shape, causes things to work differently. In this video, we'll illustrate that. I'm not going to play the whole thing because it's a bit slow, but I'll kind of show you a few things because I think it's useful to visualize the shape changing. And whoever made this for some reason didn't make the video the way most people do. So this is the inside of the cell. This is the outside of the cell. Usually you have it the other way around, but it doesn't really matter. So let's see what happens. And I don't think this one has audio, so I'll fill in with my wonderful audio. Okay, so here you have a binding site, and now you have ATP coming in, energy source. ATP provides energy for so many things inside of you, all right? So now you've added energy to a situation so you can get the proteins to do something they couldn't normally do because it's energetically unfavorable to do it without the energy source. So now what's going to happen here is that you have potassium ion binding sites, and there's this, you see how this movement is happening? Basically there's a shape change, and that shape change allows these potassium ions to now come in from the outside of the cell to the inside of the cell. After that happens, there's a change in the conformation again to a different shape, and it's kind of like the door is now, it was open this way, now it's kind of opening this way, and now instead sodium ions are going to come in, or go from the inside of the cell to the outside of the cell. And they're doing this because they're being trapped by this channel or this pump, and then using energy, they're basically being kicked out the door in both directions. So that's kind of how it works. I hope that helps a little bit visualizing. You basically have these protein domains, and they're basically changing their shape to push things in or out. This one is active, and this one here was passive. Okay. Yes. Yeah. Yeah, I believe so. I'd have to go back and double check. But yeah, the question was, is the ATP being converted basically to ADP or AMP, right? Are you using both groups? And I believe this one does. I think it's like a two-step thing. So it uses some energy and some energy again. Yeah. But I have to double check that. But I think so. Okay. So with that said, any other questions on ion channel coupled receptors? That's really it in terms of that class of receptors. Okay, GPCRs. This is the one that's really dense and that we're going to spend a ton of time on. So, as I already mentioned, these are 7-pass transmembrane receptors, so named because 1, 2, 3, 4, 5, 6, 7. This is what they look like. They've got the outside of the cell where something can bind to it, and then inside you've got these loops that can interact with different proteins to take the signal forward. Do you have a question? Yeah. Oh, okay. So what does coupled mean? Coupled, that's good. So coupled means when something is interacting with or stuck to something. So coupling is a general term. And in this context, they're called G-protein coupled receptors because of what you'll see next. You're going to see that when a signal comes through, that signal is directly coupled or sent to nearby G-proteins. So we'll get to that in a second. So some examples of ligands, of agonists that activate GPCRs are here. And there are many others, but this gives you a sense of how broad it is. When you smell your coffee or your pizza, you're using a GPCR. When you respond to light, when you respond to, your cell responds to chemoattractant, there's a ton of examples where GPCRs are used, and there's a bunch of different ones, and they do different things. This is a big family of, like basically, the title of these last couple of lectures has been, how do cells respond to their environment? Well, this is the number one answer if you were playing Family Feud or something, GPCRs, all right, at the very top, okay? So here we go. What we're going to do next is look at the mechanics of how this works. I'm going to first play a movie for you that summarizes the process. Don't freak out. It's a lot of information, but we're going to then go through the movie step by step, rather the information step by step. But the movie does a really nice job of giving you an overview. And this, by the way, is why reading chapters beforehand is great. If you read up on GPCRs before right now, you're going to find this a lot easier to do. So if you're not doing it yet, please read your book chapters before coming to class. All right, so let's play the movie.
Many G-protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G-protein for short. As the name implies, a trimeric G-protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G-protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G-protein is inactive. In some cases, the inactive G-protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G-protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G-protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G-protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G-proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound.
Okay, so that is G-protein coupled signaling. One of the first points I want to make from this, to recap, is that this is using that molecular switch, one of the 2 I told you about just a few slides back, where you're using GTP and GDP and switching out that phosphate group or switching out the molecule itself. And the light switch analogy I mentioned earlier, with the light being on or off, in what happens with GPCRs, quite deliberately, is that light switch is being flicked up and down constantly, like a little kid just won't stop playing with it. And that's deliberate, because what it does is it finally regulates the amount of signaling. These pathways are so critical that you don't necessarily want something to be always on or always off or for too long. And so by having this be a quick on, quick off thing, you can really carefully send a signal in certain amounts over time. And we'll go through that in more detail. This movie I highly recommend watching again to study. And again, it's in lecture capture, Echo 360, and you can take notes on it and all that good stuff. And this diagram here summarizes what we just talked about. So let's go through it as a quick overview, and then we'll go through all the details. Heterotrimeric G proteins. Hetero means different, right? And trimeric, tri for three. So 3 different proteins. Heterotrimeric G proteins are 3 different proteins. Alpha, beta, gamma. Very creatively named, right? Alpha, beta, and gamma form a complex. That's what you saw here, alpha, beta, and gamma. And when that complex is bound to GDP with the 2 phosphates, diphosphate, it's off. It's that molecular switch I showed you about. It's off. What happens here is that when a signal comes through, and this goes to your question, this is why it's G protein coupled signaling. G alpha is coupled to what this receptor does. The receptor basically stimulates the removal of the GDP, as shown here, and the addition of GTP instead. That turns it on. It's the molecular switch being turned on. So this is the switch, and your receptor is what's turning the switch on. Once that happens, this complex is active. Once it's active, it quickly dissociates, comes apart to alpha with the GTP still bound to it, and beta gamma on their own. And now both alpha and beta gamma can signal because they're free to do so. Their shapes have changed. Whether or not beta gamma signals depends on the context. Some receptors it does, some receptors it just hangs around. But to my knowledge, G alpha is always signaling once it's like this. This is the part where the kid flicking the light switch happens. As soon as this is turned on like this and breaks away, within sometimes, certainly within seconds and sometimes even faster, it turns right back off. So that it has just a moment to send this signal forward through the signaling cascades we're going to talk about. And then goes back to being, just one minute, to being the off state. And then when it's got the GDP now, because the phosphate came off, that can now bind to the beta gamma and form the inactive complex. Yes? Sorry, say that again? That's a fantastic question. Do G proteins, so the alpha, beta, gamma, do they come from the cytoplasm or the cytosol, or are they attached to the membrane? The short answer is it depends. But what they do tend to do is hang out very near the membrane. In many cases, and I'll show you in this video, the G alpha is actually directly attached to the membrane. And perhaps that's always the case. So this pink squiggly line indicates that G alpha is attached. It hangs out in the membrane, and it can slide along and concentrate. You've probably, I think, learned about bilayers and how proteins can move in those bilayers pretty quickly because it's sort of like this pseudo-solid liquid kind of a thing. Now here, they're showing a pink squiggly thing for the beta-gamma as well as in the gamma is attached here. That can also happen, but I think there are some cases where it's free as well. Is that correct? Yeah, right. So it really depends, but I believe the G alpha is predominantly found attached to the membrane. Yes?
Two questions. Does one signal produce one of those tri-proteins turn to GTP per signal, or does one signal do the two? 100 of them, or is it signal? And what happens to the signal?
You guys are asking good questions here. So the question was, is one signal causing this to happen for one, let's say, G protein complex, or is it one signal doing this for a bunch of them, and then what happens downstream of that was the second part of your question. So, oh, what happens to the signal? Okay, so then what happens to the signal after that? So the first answer to your question, or the first part of your questions, is that one ligand binds to one receptor. And so what we're showing you downstream of here is what's happening downstream of that single receptor. But of course, in reality, in a cell, things aren't working that way. So you may have hundreds, maybe even thousands of these receptors on a cell surface. They're often concentrated in different ways. That's how, remember the videos where cells are moving towards something like a signal? That's often because they've got more of the receptors that respond to the signal on one side of them than they do on the other. So certainly the distribution of the receptors is quite variable and dynamic. The response for a receptor is to a signal. Now, once the signal binds and activates the receptor, in most cases it falls away, leaving the receptor now available to be reactivated. And again, that's deliberate, as you want to quickly turn it on and then have the ability to turn it off. And if you found a signal that stayed bound to your receptor, you'd keep that light switch on too long. In fact, I'm going to mention a couple examples of that down the road. So, yeah. Yes? They can. So we won't get into that level of detail in this class, but beta gamma can and does often signal as well. So you could think of it as like a 2-for-1 deal. Sometimes the signal comes through to the receptor, and now the alpha is going to go do this thing, and maybe beta gamma goes off and does something entirely unrelated, but serving a similar purpose. Other times, beta gamma just hangs out, does nothing, and waits for the alpha to rejoin it. Okay. Yes? Yeah, so we're going to cover that. It's being hydrolyzed, and we'll talk about that in a second. Yeah, absolutely. Okay, so the second part of the overview of this is what happens downstream of here. So you've got this binding that happened. You've turned it on. You've turned it off. What happens next? What happens when you let your activated alpha with the GTP bound to it? There's a couple of different downstream pathways. In this class, we're going to talk about this one, the one I'm showing you here. It's the adenylate cyclase-based pathway. There is another pathway that I will briefly mention to you later on, but for time purposes, we're just going to go through this one. So this pathway works through an enzyme called adenylate cyclase, also known as adenylyl cyclase. So if you ever see the ATE here, and you will in fact in some of these slides, replaced with a YL, adenylyl, it's the same thing. And you will find both used all the time. And so that's just the way a little bit of a difference in nomenclature, but it means the exact same thing. So before I go through it, I'm going to show you another video that shows, and then we'll talk about that the same way we just did.
Adenylyl cyclase is a membrane-bound enzyme. Catalytic domain is activated by the GTP-bound form of the stimulatory G protein alpha subunit, or G-alpha-S for short. Activated adenylyl cyclase converts ATP to cyclic AMP, which then acts as a second messenger that relays the signal from the G protein-coupled receptor to other components in the cell. In most animal cells, cyclic AMP activates cyclic AMP-dependent protein kinase, or pKa. In the inactive state, pKa consists of a complex of 2 catalytic subunits and 2 regulatory subunits. The binding of cyclic AMP to the regulatory subunits alters their conformation and liberates the catalytic subunits, which are now active, and phosphorylate specific target proteins.
Okay, so all of that, which is not, it was pretty brief, is shown right here. R means regulatory subunit, C means catalytic subunit. Here's your protein kinase A, or pKa. It's inactive, hanging out, not doing anything in this context. And what happens upstream of that is that, so see I use the word upstream, right? So we're talking about pathways. Upstream of that in the pathway is when you have this activated G-alpha, it takes it makes this adenylyl cyclase, it basically activates it. Once it's active, because it's bound to this activated G-alpha that bound to it, and the cyclase is shown here in purple, then that in turn takes adenosine triphosphate, ATP, and turns it into cyclic AMP. And that cyclic AMP then binds to this inactive protein kinase A and turns it on. And then it does a bunch of other stuff. And we're going to cover this again, but this is the overview. All right? So very quickly, just to mention, once you become familiar with pathways, you can even interpret diagrams that are poorly or incompletely labeled. So you can kind of test your skills so far based on what I've told you. Here's a hormone. It seems to be maybe binding to this red thing, and then something's happening downstream of here. What do you think this yellow one is based on what you see? G alpha, right. So you can see these shapes, and with incomplete information, I never labeled this G alpha. You can tell that this is going to be, okay, that's probably G alpha. And then you can kind of figure out, oh, you know, we haven't really gotten into this much detail yet, but okay, here's cyclic AMP, and you can kind of figure this stuff out. So I'm not going to obviously do that to you. I'm not going to incompletely label a diagram, but it illustrates that you are actually learning this information right now, and that if you practice these things, it'll become more and more clear. Now, why do you have this complicated signaling pathway setup? Why not just send a signal into the cell from a receptor and say, go do your thing directly? This is one of the reasons why, this slide. Efficiency and scaling up, economy of scale. The idea that a single ligand molecule, 10 to negative 8 molar concentration, a single molecule can activate, in this case epinephrine in blue, can activate a single receptor, and that receptor in turn can activate 100 G alphas pretty fast, and amplifying that signal. All right? And then if you're activating the signal, or you're amplifying the signal through these different enzymes, each of them can go and phosphorylate or dephosphorylate or whatever it needs to do to another bunch of molecules, and each of those can do more things to another bunch of molecules. It's like rabbits breeding or something, right? Or a pyramid scheme. And all of a sudden, you've got, I know I have weird analogies. And so you end up with a ton of amplification of your downstream effect. So really quickly, with a single signal or a few signals, you can get a cell to do something quickly. It's very efficient, and that's why these pathways are quite helpful for a cell. So let's go back now to understanding this G alpha, beta, gamma stuff a little bit more clearly. I've given you the overview. I know it's a little confusing. We're going to go through it in a little bit more detail. So here again is your receptor. Here's your alpha, and then your beta is this one, and gamma. Again, notice that in this example, alpha and gamma are both tethered to the membrane. Beta is not. It's kind of squished in between. So when the signal is coming in, the receptor is directly changing the shape of G alpha to open it up. That causes the GDP to come out and the GTP to be put in instead, leading now to an activated G alpha that then moves slightly away and can do its signaling as these little lines indicate. And the beta gamma is now freed, and it's able to do whatever it needs to do. So, so this is the putting on the hat with the feather instead of changing your hat. You've activated it. All right, so conformational change means shape change. You are conformationally changing and activating G proteins because of this exchange. Now, a little bit of terminology that, and I'm going to pause before I tell you about the terminology and point out that there are these terms that can be really confusing because they often all have a G in them. You're going to be learning here about GTPases, GEFs, GAPs, and it can get confusing. But if you know what they stand for, the names actually tell you exactly what they do. So in this example, so in the first one of those terms I'm going to introduce to you, a GEF, or GEF, that stands for guanine nucleotide exchange factor. Now, if I had all of this covered up, and you've never seen it before, and I told you I'm going to use something called a guanine nucleotide exchange factor, if you thought about what these words mean, you could probably figure out what it does. It's a factor that exchanges nucleotides. Which nucleotides? Guanines. All right? So what is it doing? It's taking a GTP and a GDP, and it's switching them out. That's exactly what we just saw happen. So the receptor is a GEF. And to repeat that again, the receptor, the G protein coupled receptor, is directly acting as a GEF, because when the receptor is activated itself, it is then changing the shape of the G alpha directly to switch out the GDP and the GTP. All right? So that's what makes it a GEF, because it's exchanging these, it's acting upon the G alpha to exchange those nucleotides. G alpha, and one of you brought this up earlier, G alpha is a GTPase. All right? Enzyme, ASE enzyme that acts on GTP. To look at what that's doing, we have to look down here. Here's your G alpha. It's got its GTP attached. Remember I told you it's a really quick process of on and off. So now that the G alpha is on, it's going to turn itself off by removing the phosphate group from the GTP and turning it back into GDP. That's that molecular switch circle we talked about. Right? So I'm kind of repeating myself on purpose, telling you this a few different ways so that hopefully whatever works for you, you kind of get a sense of it. So once that happens, once this goes back to GDP, which is not shown on this slide, then this is off and it binds back to the beta gamma. And because G alpha is what is directly acting as an enzyme to remove the phosphate group from GTP, that is why G alpha is a GTPase, an enzyme that acts upon GTP to remove its phosphate group. All right? And there's another class of protein called GAPs, which are GTPase activating proteins. We're not really going to cover those in detail, but it goes back to this idea that I mentioned that signaling pathways don't work in isolation, and there's often reasons for a cell to push a reaction in one direction or the other. So this is an example where you could encourage things to turn off by getting this GTPase to work better. And so a protein that did that, a GTPase activating protein, is a GAP. That's all it means. So again, if you have these, if you know what these terms mean, starting with the basics. In other words, if you know what GTP is, and you know what GDP is, and you know what it means to activate or to exchange, you can actually figure out what these different things are doing because the name tells you what they do. So again, just for a recap, this is the molecular switch I showed you before. This is exactly what we're talking about. Before I had it very generically. Now we've filled in some of the blanks, right? So to repeat what I just told you, a GEF, a guanine exchange factor, and in this example, what is the GEF again? The receptor. The receptor is a GEF that is taking the G-alpha, a generic green protein here, and is switching out the GDP and putting a GTP in, turning on that generic green protein, in our example here, G-alpha, and then once it's on, it wants to get turned off, and you can have a GTPase activating protein, a GAP, come in and encourage this G-alpha, which is a GTPase, to act as a GTPase and to remove the phosphate group and to turn itself back off. So that's a lot of stuff, but that's why you have lecture capture and note-taking. So go back and review this stuff, and it'll start to make sense, all right? Sorry?
You said GDF is a signal, which is the receptor telling the enzyme itself to gain to the GTP, and then you said it hydrolyzes itself to loses it, and it's the GAP that helps it do that. Is that a signal within itself, or no? That's
so is the GAP, the GTPase activating protein, a signal? That's kind of like one of those semantics kind of things. I mean, you could call it a signal if you want to, in the sense that inside of the cell, you've got this other protein called a GTPase activating protein coming in and encouraging things. So in that sense, it's a signal. But it's not a signal in like the sense that we talk about ligands binding to receptors, right? Because this is all happening inside of the cell. So yes, it's a signal in a very broad sense. But you see that, yeah. Any other questions? Okay. Since we are a little bit more than halfway through class, it's time for the Twitter science break. That seems like a good time to do that. I see some fading attention spans over there, some glassy eyes. So again, how do cells learn from their environment? Dylan Burnett's over at North Carolina. He does a lot of imaging, similar to what I do. And I like some of his Twitter posts because he really gives these cells personalities. This is a story about two fish keratocytes. I like going right. Well, I like going left. No, I think I'll go right forever. Nope, you're coming with me. Where are we going? Probably somewhere a lot like here. And so let's play that and take a look at it happening. These are two fish cells in a dish, basically, keratocytes. This cell's going that way. It runs into this one. Presumably there's some cell signaling going on here saying, no, hold on, come with me. And of course it grabs onto it, and now it changes its direction. And now they're going somewhere else just as unexciting as where they were. And in fact, I think he has a follow-up or dialogue that he has down here. If I can, oh, I know one. Whatever. I'd have to find it again. Anyway, so that's our little Twitter break, looking at cell signaling with its environment, and your environment can include other cells because they will tell you what to do as well. Okay. With that, we'll get back to the not as exciting slides. But what I want, what you can kind of think about is that what I just showed you, those 15 or so seconds, that's because of the GPCRs. Like all these details you're learning about, that's how those cells are responding to each other. And not just GPCRs, there's other receptors, there's other ways of signaling, but guaranteed there are GPCRs active that tell that cell, wait, no, I want to go this way instead. Okay, so so far I keep talking about G proteins as activating, right? The G alpha is activated, it's going to send a signal. There is actually a little bit of a twist to that. There are different types of G alphas. Some G proteins act as stimulators of signal transduction. They're called GS for signaling, S for signaling, or stimulation. I think it's probably for stimulation. So either way, however you want to remember it, GS stimulates signals to move forward. Yet other G proteins, G alphas, are GIs, I for inhibition. And they can actually stop the signal from going forward. So now, the light switch analogy, sometimes maybe you're turning the light on, but the end effect is something inhibitory, even though the lights come on. And that's kind of the way maybe to think about it. This side of this diagram, so here's adenyl cyclase, that enzyme that I mentioned to you is the next step in this pathway. So it's sitting over here in the membrane. This side of this diagram is what I've already told you. There's a stimulatory ligand that's going to activate this receptor, and it's going to then activate this G alpha beta gamma complex, and then it's going to go ahead and act upon this enzyme. We've already covered that. Here's what's going on when you have a GI. So the difference is right here. Sorry for the resolution, but basically this is GS alpha, and this says GI alpha. And by the way, you're going to see this written a few different ways. People aren't consistent about this, unfortunately. It all means the same thing. So if you see GS alpha, that's the same thing as GS. And if you see same thing as G alpha S. Basically, if you see an S, it's stimulation. Same thing here, I for inhibition. So what's happening here is the initial part of this is the same. You have a ligand binding to a receptor that's going to activate a complex. But notice that the ligand is different. Okay? So in this case, the ligand is trying to inhibit the downstream signaling, not activate it like you saw here. And because the ligand is different, and the receptor in this example is also, you may notice, different, beta 2 versus alpha 2. The receptor in this case is going to activate GIs, not GSs. So the mechanics are the same, where the GDP gets exchanged for the GTP and all the rest of it, and the alpha separates. All that's the same. But the end result is the opposite of what this GS would do. Because this GI alpha will bind to this adenyl cyclase and turn it off. All right? So there are different types of G alpha based signaling, stimulatory and inhibitory. Now, there are two other types of G proteins, two major other types. I'm not going to cover them in class, but you should be generally aware of them for your entrance exams. If you ever see it, don't wonder what on earth a GQ is, other than yeah, anyway. So GSs, again, they stimulate adenyl cyclase. GIs inhibit adenyl cyclase. And then where it gets a little bit more complicated, I'm not covering this in this class, is that GI can actually activate phospholipase C in a different pathway. So it gets even more complicated. I'm not going to cover the phospholipase C pathway, but when you study for your exams, you may want to go ahead and brush up on that a little bit so you're aware of it. And then there's GQ, again, usually through phospholipase C, and G12. And this shows you how little we know, how much we're still learning. We still don't know what G12s do. They exist, they signal, but we don't understand much beyond that. Go figure. So we're going to focus mostly then, we're focusing in this class on GS, GI, and on the activation or inhibition of adenyl cyclase. Okay. So again, to recap some of this and to show it to you yet another way, G alpha, here it is activated, right? So we kind of stopped in this diagram back here. Here's your activated G alpha. What happens next? I already kind of covered it. I'm going to recap it for you again, is that activated G alpha, it's going to turn itself off by hydrolyzing its bound GTP. It's a GTP ace because it's going to hydrolyze its bound GTP and turn itself off. So here you have it activated. If you follow this through, you're having this phosphate group removed from the GTP. The alpha in this light brown color is taking it off. Once that falls away, now you have GDP bound to your alpha, and it immediately goes off. Now that it's off, it needs to rejoin with the other subunits, the beta and the gamma, and those come together. They're floating around, and they pretty quickly find each other. Oh, okay, you're in that sticky conformation now where you're off. Let me join with you again and make my heterotrimeric G protein complex. And so that's what happens, and now you have it inactive again. And the process can repeat if you get another signal coming through here. So again, on, off, on, off. Now, taking it to a little bit of health relevance, you may have heard about some toxins, cholera toxin and pertussis toxin. These are some nice examples of how things can go wrong if you keep things on or off for too long. Cholera toxin, it can be quite deadly because what it happens is that it binds to and stops the G alpha from hydrolyzing its GTP. In other words, the G alpha here stays bound to GTP, which means does it stay always on or always off? Say it again? On, right. And if you're signaling through this pathway, whatever you're doing downstream is always on now. And that can be really bad for a cell. I mean, there's you can think about a lot of examples where that could be bad for a lot of different ways, whether you're stimulating cell division, apoptosis, growth, whatever it is. Anything that's done beyond what's supposed to be happening is a problem for a cell. And if this is happening throughout your whole body, you're going to have a lot of cells failing at what they do, and then you can have complete shutdown of your body. Pertussis toxin, just as dangerous, does it a different way. It binds to this G-alpha I and stops it from shedding its GDP. So if you can't get rid of your GDP, you're staying always on or always off? Off. And again, this goes back to the idea that on-off states are forks in the road, where each state is really, really important and has to be carefully regulated. So just having something off all the time isn't okay. So when it needs to turn itself on, it won't be able to. A couple of real-world examples of the relevance of these things to our health. Okay. I'm going to bring the two types of receptors back together in a sense. These things are not happening in isolation. And one example of things coming together is with a cooperation between GPCRs and ion channels, the type of receptor I covered in just those two slides. Acetylcholine acts on heart muscle cells as well as other cells. And on heart muscle cells, it slows down your heart muscle contraction. So it kind of chills things out in terms of how fast your heart is beating. And acetylcholine does this in an indirect way, if you will. It binds to a GPCR and activates this G protein-based signaling that we've talked about. And this G-alpha, once it's activated, it goes off and does its own stuff. But this is an example of the beta-gamma actually doing something as well. And what the beta-gamma does is it floats away and binds to a potassium channel and opens it up. So it is basically, if you will, acting as the ligand, in a sense, binding to a receptor, causing a shape change to then change what happens downstream of it. And what happens downstream of it is that these potassium ions flow out of the cell, from the cytosol into the extracellular space. Now, why does this affect your heart? Well, your heart contracts based on differences in ion concentrations that create a membrane excitability that helps it drive in a quick burst contraction of the heart. We're not going to get into detail. This is not like a cardiac physiology class, but that's a very broad level description of how a heart muscle cell contracts. And if you reduce that ion gradient, if you had a bunch of potassium here and none out there, and now you've taken that gradient and you've reduced it somewhat, your cell is not going to, when it tries to contract, it's going to contract more weakly or more slowly. It depends on what it's doing. So you have reduced the membrane excitability, and now the heart muscle is going to contract more slowly. So again, it's a GPCR sending a signal then to an ion channel. Epinephrine, otherwise known as adrenaline, works on a different GPCR called a beta-adrenergic receptor and does the opposite. It gets heart muscles to contract more quickly, more rapidly, that flight or fight response. So these are a couple of simple examples of how you can have ligands acting on GPCRs, and there are so many different ligands and GPCRs that you can have different types of effects based on what you need at a given moment. So I kind of already went through this, but some G proteins activate membrane-bound enzymes. So here's an example with this beta-gamma complex that's activated, and then you have an alpha that's activated here. It's binding to an enzyme at the cell membrane. We covered adenyl cyclase. That's an example of that. And then it has a lot of signaling downstream of that takes place. Now, the next few slides I'm not going to test you on, but I again want you to be aware of them for your entrance exams. These are some common effectors downstream of GPCRs. I don't see any value in having you memorize them, but you will need to be generally familiar with them if you want to take some of these exams and for your careers as well. So there you have it, lecture captured. You have the PDFs. These are some common intracellular second messengers. Again, we're not covering them with the exception of cyclic AMP, which I am going to be talking to you about and already did, and a little bit on calcium. So now, yeah, so those are the two slides. I'm not really focusing on them, but just be aware these things exist. Okay, so now let's get back to our pathway then in the last 15 minutes or so. And we're going to focus on cyclic AMP. So remember, adenyl cyclase is being activated by these GPCRs, one of the major pathways that's activated. And what it does most immediately is remember it's membrane bound, as I mentioned, and it's going to take adenosine triphosphate, ATP, so here you have your 3 phosphate groups, and it's going to convert that into something called cyclic AMP. And this was in the video that you saw, the second video I showed you with what adenyl cyclase was doing. If you have any difficulty remembering cyclic AMP, I mean, just kind of think about, what it's doing here is it's removing 2 phosphate groups, and this long stretch of phosphates, 3 phosphates, now it looks kind of like a circle, like a cyclic kind of a look to it. So this is your organic chemistry or your basic chemistry right there. So adenylyl cyclase removes the 2 phosphate groups and makes cyclic AMP. I'm going to get into that in more detail, but I want to mention to you first that there's another enzyme called cyclic AMP phosphodiesterase that you need to know. And this phosphodiesterase is not membrane bound, it's hanging out in the cytoplasm, cytosol. And what it does there is it takes the cyclic AMP and converts it into AMP. So it basically gets rid of the cyclic aspect of it, if you will. And you can see the difference in structure here. All right, hold on to that thought because we're going to come back to both of these in a second. Let's talk about adenylyl cyclase first. So once you make your ATP into cyclic AMP, what's happening? Well, here it is more in visual form at the membrane. And you've got adenylyl cyclase with these domains. These integral membrane domains are present here, and you have a catalytic domain on the cytoplasmic side of the plasma membrane. Catalytic domain, catalysis is encouraging a reaction. So this is the domain, the catalytic domain that is responsible for this, ATP to cyclic AMP. Same thing I showed you here, ATP to cyclic AMP, just showing a different way. So this happens super fast, and that's important. We keep talking about how quickly you have to do GPCR-based signaling. This is an example of how quick it is. Remember, you're making cyclic AMP, right, after your GPCR has activated this? Well, here's an example where you add serotonin as a ligand that binds to a receptor on top of the cell here. And the colors indicate, the warmer colors, the reds, indicate a level of cyclic AMP based on a fluorescent output. In 20 seconds after you add this serotonin, you have massive amounts of cyclic AMP in this cell. All right? And the reporter is a protein that fluoresces when it binds to it. So it just sort of illustrates the time scale of this. You put your signal on, binds to the receptor activates G-alpha activates adenylyl cyclase, and you see it this quickly, the cyclic AMP that it makes, 20 seconds, often even faster. Now, again, this is from that video, and we've covered it, but just to recap again, the main target of cyclic AMP, once it's made, once you make that cyclic AMP from the adenylyl cyclase, the main target of that is going to be protein kinase A, which is a serine threonine kinase. pKa, protein kinase A, the K for kinase, phosphorylates. Serine threonine kinase just means that it phosphorylates the serine and threonine residues on different proteins. So the way this works in visual form is that you have cyclic AMP that's being made from this adenylyl cyclase-based activity, and that cyclic AMP is going to bind to these regulatory subunits on protein kinase A. This thing here, this whole thing is protein kinase A. Once the cyclic AMP binds, it turns on the protein kinase A. And it turns it on by this binding causing, as usual, a shape change. And in this case, that shape change, like you can see this is a circle and then this is more of a diamond, causes this to fall away. And these catalytic subunits of pKa that were previously inactive are now freed up and available to go do what they need to do, which is to phosphorylate on serine and threonine residues. So to put some of that together, oh, not yet. Any questions, by the way? I haven't asked that in a while. No questions? All right. I know it's not the most exciting stuff, but it is important, so hang in there. We'll get to more exciting things in the next few classes. Unless you just really find this exciting, which is great. Good for you. Okay. So here's an important point that I already mentioned when I talked about those toxins, right? Pertussis and cholera toxin. The idea that things can't just stay always on or always off. You have to have some regulation. In a normal state of affairs with this pathway, this adenylyl cyclase, cyclic AMP, pKa-based pathway, you don't want to keep it on all the time. So what can you do about that? Well, a few slides back, I showed you this. Adenylyl cyclase makes the cyclic AMP, and I mentioned to you to hold on to the thought that cyclic AMP phosphodiesterase is a different enzyme that then turns cyclic AMP into AMP. Now we're going to talk about why that's important. Here is what you've already learned. Whether you're stimulating or inhibiting, you have G-alphas, G-alpha-S, G-alpha-I, that can act in different ways on adenylyl cyclase, either activating it if you're a G-alpha-S, or inhibiting it if you're a G-alpha-I. And then downstream of that, you're making that cyclic AMP we just talked about from the ATP, which then is activating your protein kinase A, right? This is what we've already talked about. Here's where the phosphodiesterase, this guy, fits into the equation. At this stage, if you don't want cyclic AMP to be signaling too much, you can have that cyclic AMP phosphodiesterase in the cytoplasm come in and convert that cyclic AMP into AMP, just like shown here. Once that happens, the process dies. You've basically put a dam in the pathway, going back to the analogy of rivers flowing and either going downstream or having something that stops the process. In this case, you have inhibited the process from moving because you have taken all that cyclic AMP and gotten rid of it by turning it into this. Once you do that, the protein kinase A can't be activated, so it turns itself back off. So this is, if you remember the insulin and the glycogen diagram and how there were multiple ways that things were being turned on in both directions, whether you're making glucose or getting rid of it, the idea here is kind of the same idea. You want multiple ways to turn pathways on and off so that the cell is able to finally regulate the levels of the downstream signaling. All right. So how are the effects of a single signal coordinated among different cells in the organism? We already talked about this a little bit when we talked about the same ligand binding to receptors and causing different responses. If you remember the slide with salivary glands being stimulated to secrete saliva versus a heart cell being stimulated to do something else versus blah, 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 right? So one of the ways you coordinate a single signal is by having different downstream responses. And this slide, which please don't try to memorize this, is an example of that, just to give you a sense of how diverse and broad our responses are to these signaling mechanisms. These are all examples of hormones here, and where they come from, and what they do. So, for example, testosterone starts in your testis and is a steroid that induces and maintains male sexual characteristics throughout your body, right? Glucagon is from the pancreas, but it does things throughout your liver, your fat cells. So these are examples of signals that have to do different things across your body in different ways. One of the ones we'll talk about in detail, again, as an example, which you've already heard, is adrenaline, the flight or fight response. This guy's got the right idea. And this adrenaline is acting, again, to take it, this, hashtag throwback, I'm taking it back to an earlier slide here, is an example of endocrine signaling. Right, so I'm trying to integrate concepts for you, and I hope it helps, and definitely go back and look at this, is that these are examples of endocrine signaling, where you're starting in one location and going into the blood and going to other parts of your body and doing something. So now you kind of have a better idea what that actually means. In the case of adrenaline, stress of some sort causes the release of stress hormones from your adrenal glands, which are down here. And these hormones include adrenaline as well as other things that are released. And they have to act all throughout your body, including, of course, on your brain to let you know what's happening here. These are some examples of the receptors that bind adrenaline. Again, I don't want you to memorize these different receptor types. I want you to take away the concept. And the concept is this. Notice something. These are 4 different types of receptors, grouped into 2 subcategories, and then, the four different types, alpha, beta, 1, 2. And these are the effects that they have in different cells upon stimulation with adrenaline. Compare what they're doing. Okay, inhibit insulin, that means more glucose. Glucose metabolism, that means more energy. Constrict my blood vessels, that's going to, bring my blood pressure up. Increase my heart rate. All of these things feed into smooth muscle relaxation in your lungs so you can breathe better. So you see how these different responses all come together to make your flight or fight response. What happens for you in a split second when you're reacting to a situation where you have to do something, whether you're us or an organism that needs to get away from a predator or catch its prey or what have you, all of these things are coordinated in very short amounts of time because of the differences in cell signaling that are specific to different types of cells. So that's kind of cool, right? I mean, I know the details are really, you have to learn them, but the end result is fascinating when you think about it. How inside of all of us at every moment there are these multiple things happening that make us function. All right? So in the last few minutes, I'm going to round things out here, and you will get out on time, I promise. Adrenaline, as I mentioned, is acting on different cells to prepare the body for action. This is one example of that, glycogen breakdown. And the cyclic AMP effects that we've discussed, they can be rapid in seconds or slow. So again, this takes us back to that idea of both slow and fast responses, that you can have a quick response in seconds, but then you may also want to modify your cells over time. And this activated protein kinase A shown here can go into your nucleus, and in many cases will actually change the transcription of what RNA is being made. So slow and fast responses put into context for you. based on the effect of adrenaline. And then I won't go into it in detail, but I find the NobelPrize.org website to be a fantastic place to go and read because obviously I'm super cool like that. And joking aside, if you go and read about Nobel Prizes that have been given out, their summaries are some of the best examples I've ever seen of how to summarize major scientific advances. They're summarized by people for a lay audience. So if you find any of this interesting, this is an example. The Nobel Prize in 2012 in chemistry went to these two gentlemen for understanding the inner workings of G protein coupled receptors. And I also want to mention this because 2012 is only 7 years ago, right? Now, they did the work way before that. You win a Nobel Prize after decades of work. But it gives you a sense of how we're still figuring this stuff out and how important it is to keep doing research and to understand how things inside of us function. So getting back to this then, I'd like to mention a clinically relevant thing that many of you have had to deal with, and maybe your friends and family have had to deal with: smooth muscle relaxation. Asthma is very prevalent in our society and has increased because of a variety of reasons we don't have time to get into. One of the wonderful solutions is that many people carry an inhaler that works really well to relax your lungs so you can breathe better. How does that inhaler work? You just learned about it today through GPCRs. So let me show you how that happens. This is a cross-section of your smooth muscle cells surrounding basically the airways that you have. And so inside of your lungs you have these bronchioles. To get them to relax so you breathe better, the smooth muscle that surrounds those bronchioles has to relax. And what happens is that adrenaline, working through an adrenergic receptor, like we discussed, is actually involved through pKa and encouraging that relaxation, which is something that was on this right here, smooth muscle relaxation. So why is this relevant to asthma? Well, if you have a situation with an asthmatic bronchiole where you're not relaxing, you're inflamed, that's inflammation is what asthma is, and you need to relax it. Long time ago, companies came up with something called albuterol, which acts as a beta adrenergic agonist. In other words, the albuterol that somebody takes, that goes, you breathe it into your lungs, it binds directly to these beta adrenergic receptors where adrenaline would have bound and has the same downstream effects, resulting in relaxation of your target cells. So this is where pharmaceutical companies or biotech companies can take these pathways and come up with wonderful products that help us in our daily lives, such as the ability to help people relax who have asthma. And then the last thought I'll leave you with is this, salmeterol. It's a long-lasting version. It's a better version. It's like albuterol on steroids, pun intended, because this is a steroid-based thing, ha ha ha. Okay, so salmeterol works even better than albuterol. Why? Well, check this out. This is the structure of albuterol, and it looks really similar here to the structure of salmeterol, right? You have the same thing going on. The difference is this long add-on. What does this long add-on do? It's lipophilic, and it binds, it grabs onto cell membranes. And so what happens is you put this guy in, you have this part binding and activating this receptor, but then you also have a piece sticking out, and that piece gets stuck in the membrane here. As a result of which, this agonist here that's binding here and activating this receptor, once it activates and releases, it still hangs out there because it's stuck. And then it binds again and again and again. So it's sort of like, hey guys, please, I don't want to, it's hard for even me to hear. You'll get out on time. Thank you. So what happens is that you now have this thing stuck in the membrane, and it's almost like a duck bobbing in the water or something, and the agonist keeps binding to the receptor and stimulating this relaxation. And that is why salmeterol works even better than albuterol does. With that, we'll stop. You're getting out a minute early. Next time, I want to hear you guys not pack up in advance, please. It's really distracting. Thanks. Have a nice weekend, and I'll see you on Tuesday.